Hello, today my group and I are going to present the topic on next generation sequencing. According to the statement, DNA sequencing is the process of determining the precise order of nucleotide within a DNA molecule, but we would like to go in depth by discussing the applications of next generation sequencing in cancer treatment. So here are the aims for today's topic. We would like to understand what is next generation sequencing is about and how does it work. We also would like to know about the differences between the old generation sequencing and new generation sequencing, as well as to investigate the applications of using next generation sequencing in the cancer treatment. Finally, we'd like to highlight on the future applications, challenges faced and recent development of next generation sequencing. So the caption for today's topic is cancer therapy in current context. First of all, cancer in its many forms has accounted for 8.2 billion of deaths in 2012, and the rapid development of the NA sequencing technologies has basically driven a revolution in our understanding of these highly complex and diversified groups of diseases. There are different types of cancer, including CML, known as chronic myelogenous leukemia, that featured a single and pathognomonic DNA mutation. By working on this assumption, early methods have been utilized in exploring the foundation of genomics of different cancers. It involves exploration of targeting specific variants and genes in low throughput fashion. Human Genome Project, which was a successful project in the early 2000s with increasing affordability and reliability of sequencing, has led to the integration of genome sciences into clinical practice. The usage of this data in assisting of diagnosis is known as precision medicine.
However, demand for revolutionary technologies that deliver fast, inexpensive, and accurate genome information started to arise. This challenge catalyzed the development of next-generation sequencing technologies. Here, we are going to highlight the importance and potential of a complete or 100% genome sequencing, next-generation sequencing. What is next-generation sequencing? It is commonly termed as massively parallel sequencing, whereby it's a way of capturing a very big amount of genomic information about a cancer. NGS came as a revolution in genomic research. By making use of NGS, a whole human genome can be sequenced in a single day as opposed to the previous Sanger sequencing technology. Sanger sequencing technology took more than 10 years to deliver the final draft of a complete human genome.
So, how does next-generation sequencing work? Most NGN technologies uses sequencing by synthesis, whereby each DNA fragment to be sequenced is bound to an array, and then DNA polymerase adds labeled nucleotides sequentially. A high-resolution camera captures the signal from each nucleotide becoming integrated and notes the spatial coordinates and time. The sequence at each spot can then be inferred by a computer program to generate a contiguous DNA sequence, referred to as a read. So as you can see here, this is a summary of the workflow for NGS sequencing in oncology. The main steps would be choosing approach, utilizing results, and continue monitoring. So first they have to consider the condition of the patient, by their tumor type, how to ease the method of sampling, and the previous workup of the patient. And based on the condition of the patient, they have to choose between different assays. It's either target gene panel, whole exome, or whole genome. Based on the assay they decide, there's different sampling methods that can be used. As you can see here, it's either FFPE biopsy, fresh frozen, or ctDNA. After sampling, the sample will be sent to bioinformatics to get the aligned genome, core sequence variant, and future artifact. So after that, the scientists will check the database for mutation hotspot, actionable variant, and prognostic value. After checking the database, only they can decide which therapy is most suitable for the patient. So here you can see targeted therapy or clinical trials to avoid unnecessary therapy. After having therapy for some time, the patient will undergo liquid biopsies to look for increase in allele fraction of identified mutations and to monitor resistance, and then they can adjust the therapy as dictated by results. So what is the benefits of using next-generation sequencing? By using NGS, the samples no longer need to be handled in a different manner from standard diagnostic specimens. This new advance enables increasingly complicated genomic data to be obtained from an individual's blood sample only. Genetic mutations have long been identified by Sanger sequencing, PCR, and microarrays. However, they have limitations that don't apply to NGS. For example, although previous methods can detect single nucleotide variants, they have trouble identifying larger DNA aberrations, such as large indels and structural rearrangements which are common in cancer. On the other hand, whole exome and whole genome sequencing can provide a comprehensive view of the DNA aberrations, genetic recombinations, and other mutations. Therefore, NGS platforms serve as a good diagnostic and prognostic tool and help clinicians identify specified characteristics in each patient, paving the road towards personalized medicine. So here you can compare between Sanger sequencing and next generation sequencing. For Sanger sequencing, it has pros of lower error rate, longer read length which is up to 450 base pair. But it can only do one sequence at a time, which only produces one forward and one reverse read. It needs a lot of DNA to start with, and it has a higher cost per base, which is more expensive compared to next generation sequencing. On the other hand, for next generation sequencing, the pros are it can produce massively parallel fragments to be sequenced in a single run. It has fast turnaround time and it is cheaper. But it can only have few reads of each basis that are combined, so less accurate overall. And it has short read length.
Hi guys, it's Madiiha here, and right now I'm going to talk about the applications of NGS. So to start with, NGS has already been applied in the clinic for cancer diagnosis and prognosis. For example, by doing the sequencing of the whole genome, researchers were able to identify a novel insertional fusion that makes a classic BCR3-PML-RARA fusion gene for a patient suffering from acute myeloid leukemia, AML. The findings brought about alterations in the treatment plan of the patient. Another example would be in oncology studies whereby NGS is used to identify informative mutations in a clinically relevant time frame. With this approach, identifications can be made on specific and individualized mutational landscape or the observation of cancer evolutions for patients suffering from metastatic colorectal cancer and malignant melanoma. Third example is using paired-end sequencing to generate a comprehensive catalog of somatic mutations in a malignant melanoma genome. And the last example is sequence cancer genomes from metastasis and primary breast cancer samples to investigate the mutational evolution of cancer cells. The second application is using NGS technology. Clinicians have the possibility of designing patient-specific probes that makes use of DNA found in the patient's blood serum to monitor progress of a patient's treatment and note any sign of relapse. Now I'm going to pass the rest of the applications to Ivy.
Additionally, next-generation sequencing is utilized by deeply looking into the sequence of targeted regions, especially the exon region, or known as target sequencing. The purpose is to target multiple candidate genes in providing a comprehensive view of the genetic makeup of the cancer patients. For example, mutations of JAK2 subtype gene in myeloproliferative neoplasm is discovered using this method. Furthermore, RNA sequencing is used in simultaneously analyzing on providing an insight on the level of gene expression, the structure of the genomic loci, as well as the presence of sequence at loci. For example, pair-end sequencing is used on adult granular cell tumors, or GCT, to compare with the transcriptome of 11 epithelial ovarian tumors and published sequence of human genome. They discovered mutation of FOXL2 gene was discovered to be involved in the granular cell development and its pathogenesis. On a side note, pair-end sequencing basically allows users to sequence both ends of the fragment and generate a high-quality alignable sequence data, particularly to references where this improves the quality of the entire set of sequence data. It also facilitates detection of genomic rearrangement and repetitive sequence elements, as well as gene fusions and novel transcripts.
Okay, I'll be talking about the treatment using NGS, which includes standard clinical practice in guiding cancer therapy, personalized treatment of cancer in clinical oncology, and pancreatic cancer treatment and non-small lung cancer treatment. Follow, we'll be talking about standard clinical practice for cancer therapy. NGS is a result of multiple technological and practical advances. Earlier protocol required outside of typical pathology workflow to harness or obtain the starting material. Now, NGS will be using standard formalin fixed or paraffin embedded specimen for the NGS procedure material. And also the knowledge base of the NGS are also being masked, which means that not only you will be doing the NGS, the others clinic will be using as well. Protocol for analysis and NGS data interpretation will allow a clinician easily obtain information for patient care. Then followed by personalized treatment of cancer. It will be low cost, shorter turnaround time, as is usually the treatment of cancer will be quite high cost and long time to obtain the result, because of its ability to fully sequence large number of mutation genes of all types in a single test. Second, more comprehensive analysis of variation, able to detect all mutation type of the target gene and even chromosomal abnormality if compared with single sequencing. And then there will be require less DNA, because only small amount can be obtained from tumor tissue, as it is not easy to obtain large quantity. This will be the advantage of using NGS. Then we follow by pancreatic cancer treatment is using the somatic alteration of the pancreatic cancer genome in the NGS. It is able to identify therapeutic option for the patient. Followed by non-small lung cancer. Exon 90 microdeletion has been reviewed by the NGS as the most frequent genetic alteration affecting the epidermal growth factor receptor, which causes the non-small lung cancer. Then we are able to respond to the treatment using tyrosine kinase inhibitor. And this is all for the treatment using NGS example. Thank you.
So I'll be talking about the recent developments of next-generation sequencing. First of all, next-generation sequencing can be used for sequencing of cell-free DNA fragments circulating in a patient's bloodstream. Cell-free DNA fragments are also known as fetal DNA circulating freely in the maternal blood circulation. By sequencing the subpopulation of cell-free DNA in the pregnant mother's bloodstream belonging to her fetus, researchers have shown the potential of next-generation sequencing as a non-invasive method for detecting Down syndrome and other fetal aneuploidies, such as trisomy 13 and trisomy 18. Based on the results from the studies, next-generation sequencing plasma-based DNA tests showed promise in detecting Down syndrome and other aneuploidies with high sensitivity and specificity. Furthermore, next-generation sequencing can also be used as a non-invasive method for detecting solid organ transplant rejection. Through the use of next-generation sequencing, increased levels of cell-free DNA from a heart transplant donor's genome can be found in a recipient's bloodstream when the transplant recipient is undergoing an acute cellular rejection. Similar to detection of a low-frequency variant, this experiment relies on deep sequencing of cell-free DNA to detect changes in a small fraction of that DNA population which belong to the organ donor. Henceforth, sequencing of cell-free DNA by next-generation sequencing in both of the examples should offer enormous potential to reduce invasive medical procedures and associated mortality. Other than that, next-generation sequencing can be used for preclinical application. By probing for the presence and abundance of microspecific DNA sequences in both tumor and normal human cell environments, the technologies themselves can be used to characterize microbial population in a culture-independent fashion. Lastly, next-generation sequencing can be used in forensic molecular biology. Genetic markers on the Y chromosome, known as Y short-tandem repeats, are most commonly used to unambiguously resolve the male components of DNA mixtures when a high female background is present, or to reconstruct paternal relationship between male individuals. With the use of next-generation sequencing, 4 genetic differences were detected when comparing more than 10 million nucleotides of the Y chromosome between 2 male individuals who share the same ancestor 13 generations ago. This suggests that Y chromosome sequencing could solve the problem of distinguishing between mixed male samples from the same parent.
What are the challenges of next-generation sequencing? First, the presence of repetitive DNA represents a major obstacle for the accuracy of read alignment and assembly. as well as the structural variation detection. Furthermore, it is quite difficult to distinguish rare mutations in tumor from sequencing and alignment artifacts, especially when the tumor has low purity. On the other hand, tumor complexity and heterogeneity make the analysis and the interpretation of sequencing data even harder. The data that derived from the next generation sequencing might take a longer time to be translated into clinical relevant information. Besides, the predictions of the functional effect and the identification of disease causal variants are still in an early phase, which means they are not well characterized yet by using the next generation sequencing. To perform the next generation sequencing, they require high performance computing with the support of bioinformatics. They also require quality control and standardization of experiments as well as the data reporting. For the future application of the next generation sequencing, they are basically applicable into the forensic studies. Besides, to move from the current descriptive and prognostic classification system to a functional genomic taxonomy, especially in cancer research. Another potential application of the next generation sequencing is to improve the diagnosis of cancer, as we know that the poor tissue sampling and processing can often make a histological diagnosis difficult. However, by using the next generation sequencing-based analysis of tissue can be performed on small amounts of viable tissue and is accurate when the sufficient information regarding to the causative mutation is known. Furthermore, they can be used to identify molecular aberration that render the tumor sensitive to certain therapies that results in exceptional responses. To facilitate clinical diagnosis and personalized disease risk profiling. And lastly, to be used as a diagnostic tools for higher detection rate in early onset diseases, such as the inherited macular degeneration. So from this presentation, we can conclude that today's complex genomic research questions demands a depth of information beyond normal capacity of traditional DNA sequencing technology. With its unprecedented throughput, scalability, and speed, next-generation sequencing enables the researchers to study biological systems at a level never before possible.
So that is the end of our presentation. Thank you all for taking your time to listen and view our presentation. If you have any questions, please post them in the SlideShare link that will be provided by Dr. Yap later on, and we will do our very best to answer them. Thank you very much and have a nice day.